Fantastic. All right. Support from our colleagues is wonderful. All right, fantastic. All right, I want to welcome everyone to our Ancillary session. We're trying a hybrid approach, so hopefully this will work. I have some folks in the room and some folks online, so that's fantastic. We have about a 2-hour session today. We have 4 wonderful speakers, but I'm just going to kind of give you an intro and start you off with some of the motivation and background. And we really want this, especially this second part, to be an interactive conversation about the resources that we're going to demonstrate for you today. So you can follow along on the slides and then pop any questions you have into the chat as we go. So I'm going to give you a motivation to start. We're going to have 2 scientific perspectives from industry and academic from the partnership that we're talking about today. And then we're going to have an overview from the portal for the portal and the Genome Atlas. So sort of 2 vignettes, but with 4 speakers. But I wanted to start with motivation and sort of why we are doing this and sort of the perspective by which we come from. We come at the perspective from a human genetics perspective and the idea that we can make target validation that much better by using human genetics focused approach rather than the traditional genetics approach. Because there are so many ways by which this is a better preclinical model and allows us to have better ways of modulation, inheritance, and mechanism in action using human genetics as your backbone, because these are experiments of nature. And so from that perspective, we want to facilitate this process from using human genetics to making better decisions about drugs, target validation, and functional follow-up of genes that might be implicated in a common metabolic disease. But the challenge is, in human genetics, we've done a really good job of identifying loci, like incredibly good job. But the challenge is, in so many of these places, we have over 100,000 loci we've identified that could be relevant targets for disease, but how is that process going to be facilitated to determine the mechanism of action, the variant, the tissue, all these questions. But what's great is that for many common metabolic diseases, we've done a very comprehensive job of identifying loci. For type 2 diabetes, there's 403 and counting with a recent paper. We have, for kidney disease, 424. So there's really a richness of human genetics data. And there's a richness of the ability to make that data accessible to the world. So some of you may be familiar with the Type 2 Diabetes Knowledge Portal, which is the first product by which our group from the Accelerated Medicines Partnership for Type 2 Diabetes developed, which made genetic data accessible, not just to a statistical geneticist, but to other folks in basic biology or in translational research to make use of this data. But other resources have done this way better than we have, right? With the GWAS catalog and the GWAS Atlas, going from a perspective of all common diseases, making genomic association data accessible. So that's fantastic. However, how do you make decisions about what gene to study and why, and what target to validate? Because in many of these cases, you see hundreds of loci, evidenced by this recent paper on kidney function, where they did an elegant job of presenting the information, but then how do you make decisions based on that? So our job is to facilitate that, to enable you to go from here to here, not doing it for you, but facilitating the process by the access to the results and the data. And so for that, the idea is a model, right? Genes influence cellular function, which influence organismal function. And conceptually speaking, regulatory elements and cell types and cell states influence that. However, experiments of nature or variation can change the regulatory elements or the cell states, thereby impacting the gene, and then ideally the cellular function and the organismal function. So our goal with human genetics is to present that information. However, you're just left with loci, right? So our goal is to facilitate that process and cultivate relationships with communities to bring together data sets that are relevant to make decisions about what is the variant, the regulatory effect, tissues, gene, or pathway, and facilitate this process with a resource. So we're going to talk about 2 resources today that enable that process. What's in the resource? So really broadly speaking, Maria's going to talk about this in more detail, and so is Parl. But the idea is it starts with genetic associations, and our goal is to make this as diverse as possible. And we now have over 400 genetic association results representing 400 traits as well, from a diverse set of countries, 64 and counting. And then the resource is also full of comprehensive diseases. So these are, not just diseased data sets, but the relevant traits and quantitative traits represented along the spectrum of those disease areas, and specifically curated by the community. Additionally, we would layer on that relevant tissue and cell type, cell-specific annotations to help you make decisions about the regulatory effect, the cell state, the tissue type, and what it's doing to the gene or the variant infection. And then finally, sort of pulling that all together, the goal is to bring bioinformatic or computational approaches, which just allow you to generate hypotheses about the molecular or cellular function using both the genetic and genomic data. So the goal is then take a software platform that brings this to you as a user and enables you to interact with it. So Maria is going to take you through that later today. So this is the portal. As you can see, anyone can log on to it. It's open access. It's cmdkp.org. And if you're interested, you can follow along with us during this session. And then the other resource we're going to talk to you about today is the cmdga.org as well, also open access. This is a supporting resource that gives you access to the genomic data that Parul will talk about. So with that, this is our mission, and what you're going to hear about next is sort of a perspective from industry as to why they want to work on such a problem to try and use human genetics to address target identification and validation. And our goal with this partnership is to really do this across common metabolic disease, inflammatory drivers, kidney disease, type 1 and type 2 diabetes, obesity, liver disease, and finally heart disease. And hopefully, because these have similar, biological pathways and underpinnings, studying all these diseases in concert will hopefully give us answers to some of the ideas for new drugs and new targets. So with that, I'll pass it over to my colleague, Melissa, who's going to talk about the perspective from industry and why this project is hopefully valuable to them.
Great, thanks. And thanks, Noelle, and welcome to everybody in the room and online. And if you were at the meeting yesterday, this will be a little bit of a repeat, so I apologize. So my name is Melissa Miller. I am a director of human genetics at Pfizer, and Pfizer is one of the companies involved in the AMP-CMD program. And so what I want to start out with a little bit is kind of talking about, from an industry perspective, how we use genetics, how we would like to use genetics better, and how the portal helps us. And so this is the two plots on the bottom here. One of these comes out of the AMP portal, the other is a locus zoom. This is a GWAS of LDL, I think from one of Kristen's original GLGC papers, and just kind of thinking back on where we were even 10 years ago with genetics. Oftentimes the information that we would have would be that we had a variant, it was associated with a trait, and we knew a p-value. Oftentimes we really did not know what the causal gene is, and this is a great example here where you can see that you have a number of different potential causal genes. Aspirationally, and I think this is true of the whole field, but it's especially true of industry where we're trying to be able to use this data to validate targets or thinking about whether we want to pursue something as a target. What we really would like to be able to say is something along the lines of, if I had an 80% decrease in sortilin activity in human livers, that would result in a doubling of circulating LDL-C levels. So this gives me a causal gene, it gives me a causal tissue, and it does give me some estimate of how much I maybe need to modulate something by. So to be able to use human genetics to come to a statement like that, there's a lot of different questions that we need to answer. From a genetics perspective, some of the questions we frequently ask ourselves, the question of SNP-to-gene or effector transcript, do we know which gene is being influenced by our genetic variant? We also are interested in directionality. Do we need an activator or an inhibitor, an agonist or an antagonist? And of course, this question comes up a lot. What is the horsepower? If I did come up with a therapeutic for this gene against a disease, would it have a meaningful impact? That's a very genetic-centric view of things. And of course, there's a lot of other different kinds of data that we often need or that often help us to be able to answer these questions. The SNP-to-gene continues, I think, to be something that we are all very interested in, and that's very challenging. There's a lot of different types of information that we use for SNP-to-gene. I'm just going to mention a couple of things here. Fine mapping, as well as epigenetic data, which can get us down to a credible set, as well as be able to start to look at where that credible set overlaps with things like open chromatin, enhancers, promoters. That doesn't always get us at a causal gene, but sometimes it can get us at a better guess of a causal gene or more confidence in which gene we think is causal. In addition to that, tissue expression is very, very important, and especially if we are thinking about things from a drug discovery perspective, we do want to know, even if I now am confident that I know my causal gene, is it expressed in my tissue of interest? Is it expressed in certain cell types? Maybe I actually want to go after a certain cell type or a certain couple cell types in a particular organ. Is it changed in disease? Is it up or down regulated? And finally, is it expressed in other tissues? And if my gene is expressed in other tissues, does that cause me particular concern if I were to try to modulate that and it hits tissues other than my tissue of interest? And the very last thing that we often think about once we've answered all these questions or answered them as best we can for the data that we have is to actually be able to perturb the gene either in a mouse model or in a cell model to be able to say, does this validate the hypothesis that I thought that it should? So AMP is a really nice resource. We use it fairly often to be able to answer some of these questions. And I wanted to give an example. This is not just exclusively data from AMP. This is data from Catalin Sustak, who was one of our AMP type 2 diabetes investigators. But I think this nicely highlights how we can use these different sources of data together. So on the top here, I just realized probably the best thing is for me to do the pointer here. We have a locus zoom plot of EGFR, which is a biomarker for kidney disease. You can see that there are a number of different genes implicated, so we don't necessarily know the causal gene. We can find map down to a set of credible SNPs and then look at cell type specific kidney ATAC-seq, and that really helps us prioritize a region that maybe we would want to knock out, which then can give us a better sense of what the causal gene is. This is a really fascinating paper in that the Sustak group then went in and knocked out a couple of different regions using CRISPR. So they did the perturbation experiment, and they were actually able to show that there are probably 2 causal genes that are acting in different directions in this locus. So one of the reasons that we really like the AMP model is because it is a great resource where we have already accumulated a large amount of this data. We have housed it in one of the 2 portals, and we can use it to visualize genes and regions of interest. And of course, part of the goal of the AMP CMD is for the diseases where we don't have all of the data that we need, for us to be able to work together, figure out how to generate that data, and to keep building the tools to be able to use this better. And I'm going to give a couple of specific examples of how we often use the portal in industry. This is a Manhattan plot from several years ago of liver fat content. So this would be a biomarker perhaps for non-alcoholic fatty liver disease. There are 2 genes identified here. One is PNPLA3, the other is TM6SF2. Both of these happen to be coding variants, and you can see what the impact of the TM6SF2 gene is there. And so oftentimes what will happen is a paper like this will come out, or we will be sitting in a meeting and somebody will say like, okay, well, what else do we potentially know about this gene? And so we can pull up TM6SF2 in the portal, and you can just take a broad view of, okay, what are the different traits that I am seeing associated with this? And of course, you can see a number of things that are liver-related, alanine transaminase, liver fat content. This is a liver enzyme. You can also see that there are lipid associations. And so from a drug discovery perspective, it's hard to know what to do with that information. If, for instance, if I were going to lower liver fat and lower lipids, that would be great. But it's also possible that I could lower liver fat and increase lipids, which is not so desirable. And so in this case, we can take that lead SNP from the liver fat GWAS, do a pheWAS on that. And in fact, what you can see is that whether I'm lowering or raising liver fat, if I go after this gene, no matter what the direction is there, it's going to have the opposite effect on triglycerides. And it's not shown on this slide, but it turns out it's also going to have the opposite effect on LDL, and it's also going to have the opposite effect on CAD. So this is a really quick and easy way to sort of say, okay, we could go after this as a target, but here's some potential safety liability or some potential safety implications we would want to think about for this gene. This is the locus zoom association for ADAM TS9, and what I have done is gone in and added in some of the tissue annotations from the portal. And so we can quickly use this data to be able to identify whether or not ADAMTS9 is perhaps has promoter or accessible chromatin in one tissue versus another. And given that you're able to identify enough specificity, you might be able to say ADAMTS9 is acting in the muscle, which I'm really interested in, but it's not acting in the pancreas. And so this would potentially be a good target if I were interested in insulin resistance. So that is one of the other ways that we use the portal, is to try to figure out if a tissue or a cell type is the one being acted on through the gene that we are interested in. All right, now I will turn it over to Struan.
All right, should we hand it over to the great Struan Grant? Beautiful.
I'll take that. Okay. Well, thanks very much for allowing me to present today. And what I'm going to talk about is, can I put this in here? Nice. Yeah, we're basically using confluence of evidence strategies to find high-value leads, and that's what I'm going to talk about. And the fact that the portal contains so many different types of data that's really going to help in that approach of finding high-value leads. Principally, what I'm interested in is GWAS signals. And so this is sort of how I see the way things are heading. When I was doing my PhD, we worked on candidate genes, and that was really the domain of molecular biologists. And then when we got into the region of looking at the genome more objectively through linkage analysis, that was really the domain of statisticians. And then when we were doing GWAS, we're very dependent on clinicians to get data to be able to get the data sets that we needed to make genetic discoveries. And then we were in the domain of sequencing, and there's a huge amount of data out there in the public domain and in the portal, and it's been the domain of bioinformaticians. So it's the idea of using all this massive amount of data that is now available, billions of dollars worth of data, and the idea of synthesizing that down to really key targets. And I would argue that the new champions are again going to be the molecular biologists who can follow up on this wealth of data that's now available in the portal and in the public domain. So this is sort of what I work on. I work on the pediatric route to type 2 diabetes. The idea being that if you're obese as a child, you're more likely to have type 2 diabetes in later life. If you've got a sort of lower birth weight, you're more likely to get type 2 diabetes, etc. But what I'd like to just raise is the childhood obesity study that we did, where we saw the top locus, the Empire State, if you will, in the Manhattan plot, is the FTO locus. And the FTO locus comes up both in pediatric obesity and also adult obesity. And as many of you probably are already aware, the FTO locus has received a lot of attention, and the outcome of that investigation has really made an imprint on how we're all thinking with respect to variant to gene mapping. So just to recap what these studies showed, there's this very strong GWAS locus embedded within the FTO gene, and the FTO gene obviously received a lot of attention. The idea is, could you have an FTO inhibitor and never have to jog again, never diet, just take an FTO inhibitor every day? It turns out that may not be the candidate gene, or it may be only one of many in the region, because these studies that I've listed here pointed to IRX3 as possibly more likely the primary culprit in the region, and also there's evidence that it's IRX5. These studies were done in adipocyte settings, but this study from Columbia also showed that there was a gene on the left. So clearly there's a tissue context to think about, and there's a sort of temporal aspect to this, is sort of which genes are in play, depending on what setting you're looking in. So the optimist would say, this is great, for the price of one signal, I'm getting 3 genes. The pessimist would go, this is really complicated. The key thing is, of course, we have a lot of data available to us now where we can start trying to tease this out and look at the context of these different signals. So really the question I think a lot of us are trying to ask in the field is, We've got a variant that's typically non-coding. It's either intronic or intergenic. Is it driving the nearest gene, or is it driving a gene further afield? And to do this, there's an arsenal of approaches that people are using. I'm not the only person trying to answer this question. And the great thing about the portal is there are these different divergent data sets in there and are going to be increasingly coming in, especially like the project I'm in that was mentioned about the type 2 diabetes project. As we generate those data, those will come into the portal. So the portal will become increasingly enriched to be able to ask questions such as what we're doing here. But the approach I've employed, and I'm generating data and will ultimately end up in the portal, is based on chromatin conformation capture. So I'm just going to talk about that. And the context I want to bring in and why I got into this area was initially to do with type 2 diabetes. So the TCF7L2 locus is the empire state building signal for type 2 diabetes, if you will. And I found it when I was working in Iceland, and I'm still smug to this day. And so this is obviously a gene that I'm very interested in pursuing and to understand what's going on. But despite my bravado about thinking I found the strongest gene associated with type 2 diabetes, is it the actual gene? Is it like the FTO locus? Have we actually worked on the wrong gene? So this is sort of what got me into this variant to gene mapping, especially on the back of that FTO study or those studies that came out. You know, the question is, am I employing too much effort on the wrong gene? And so the great thing about this gene was that it was the field has really investigated this extensively and have concluded that this is the causal variant in the gene. And the question is, what's it doing to the gene? And so what we decided to do in the very first instance, before we got into variant to gene mapping, but was just to throw a CRISPR grenade into this SNP and just say, what happens if I just blow a hole around that SNP and see what happens to the neighboring gene expression? So we made a series of different deletions around that SNP from 66 base pairs up to a KB. And this is what happened to the gene expression in the cell setting that we worked on, which was basically a colorectal cancer cell line. I'm not particularly proud of that, but we were just getting CRISPR up and running. And my working hypothesis is indeed TCF7L2 is operating through the gut for various lines of evidence that I felt supported that. The point was that we saw a diminishment of TCF7L2 expression both at the RNA and protein level. What we did see particularly strikingly in this particular cellular setting was a dramatic impact on a gene, a couple of genes away, called ACSL5. And in fact, that gene already existed in terms of the knockout mouse by Tufts, and it is indeed diabetic. So we think there's two genes operating there. This variant basically affecting the TCF7L2 expression, but also this gene 3 genes away. Now, there's a lot of debate in the field about what the actual primary genes are in this region. A lot of people are saying, Stuart, you're burning your own guitar. It's actually TCF7L2, and that's the only gene that's working there. I'm not ruling out TCF7L2, but I do think that probably because it is such a strong hit, probably more going on, and it's very dependent on cellular context. So I think that's still to be fully resolved. But certainly from our perspective, that's what we saw. So given that sort of initial study that we did, we got very much into doing more spatial and functional genomics, and Children's Hospital of Philadelphia has been very supportive of doing this, given that we'd spent about a decade doing a lot of GWAS in various different pediatric traits, and the idea of then converting those signals to the actual underlying causal gene. And this is the general idea, and these are the data sets that we're producing, and again, will eventually be part of the portal. We are finding the GWAS hits and then finding all the SNPs in linkage to this equilibrium with that SNP, and then asking which of those SNPs are open with respect to attack-seq and therefore putatively causal. So just a very simple, is it open or closed? Because normally the top SNP in a empire state building signal is not the causal SNP. It's one of those other ones in the spire. And then the additional constraint that we put on there was, is that open SNP contacted at high resolution by a promoter? So it has to be open and contacted. When we do that, we usually are down to a manageable set of SNPs at each locus that are potentially causal, and we have a lead gene. And then that motivates us to then go and do functional assays. So there's a number of chromatin confirmation approaches, and we were very interested in doing it at a very high resolution. So the box in red here is Capture-C, which is a very scalable approach and very high resolution. And we were very taken with that. That came out of the University of Oxford. And what I asked Alexander Casey and my team, who's now faculty at Penn, can we put a probe in every single promoter in the genome, along with every non-coding gene, and then have that one-stop shop where we can have looping information and then be able to say, if I've got a SNP of interest, which promoter probe is interacting with that SNP. And so we produced 36,500 RNA-baited fragments, and then we had a NovaSeq in my lab, which was very nice, and we could do very, very deep sequencing to determine these loops. And just talking about resolution, there's what's typically been used is there's 4 cutters and there's 6 cutters. And so 6 cutters been done sort of historically, and we find that you don't get the resolution with that. I won't go into too much detail, but it does have a myopia in that if the nearest gene is indeed the causal gene, really using a 6 cutter we think is probably skewed away from the nearest gene, whereas we have the possibility of determining if it is the nearest gene or further away. So that DPN2 approach, which is that 4 cutter approach, more finely dicing up the genome, is the way to go for us. So an example I'd like to use that really has motivated us to subsequently go into diabetes is lupus. And we're a pediatric hospital. We always like to cite that Selena Gomez got lupus when she was a kid, so it's a pediatric onset disorder. And my colleague Andrew Wells, who is an immunologist, said, well, if we were going to look at lupus, what would be the primary cell type that we should start off with? And so T follicular helper cells are the prime candidate. And because we see a lot of tonsillectomies at CHOP, we could sort out the T follicular helper cells from tonsils and then do our trifecta of chromatin capture, ATAC-seq, and RNA-seq. And this is one of the key loci that's receiving attention in lupus. And the LPP gene caused a bit of excitement because it was a novel gene for lupus. But we, and actually others now, have all confirmed that in fact, the variant is sitting in LPP, but it's driving a gene about 1 million base pairs away, and it's called BCL6. And BCL6 is the main transcription factor for T follicular helper cells, and it's been strongly implicated in lupus already. So what I would argue in this case, when we look at this locus, there's actually nothing new here. All we found that's new is a distant regulatory element for BCL6. But in terms of, will this target get me a Ferrari if I target it? No, because it's already well established. It's nothing new here to see. So what we decided to do was, given our data, is like, let's do pathway analysis. And so we took the nearest gene annotations, our argument being that it's not always the nearest gene. And we found that only 2 of the top 10 pathways using the nearest gene annotation were relevant to TFH cells. Whereas when we took our gene list, we found the 10 for 10 top pathways were related to TFH biology. Now, of course, we did it in TFH cells, so there's a bias there. But of course, these variants were discovered in just basically a case control setting. So we just found it very interesting that there was this enrichment. It also meant there's a lot more known biology there than we originally thought. But in there were nuggets of new biology. And that's where we're putting our emphasis and trying to do sort of some drug repurposing on novel targets already have existing compounds. Another example I like to use is bone mineral density. This is the Empire State signal for pediatric wrist. So, kids break their wrists quite frequently by falling out of trees or falling off walls. So it's either the top signal for being a knucklehead, or it's something to do with bone fragility. And so we have a few genes in that neighborhood, and the obvious gene is Wnt16, because Wints have been really implicated in bone. But our data did not point to Wnt16. It pointed to another gene, CPED1 and ING3. And what we found was that working with the University of Michigan, we found that the clear winner in our hands was ING3. It affected alkaline phosphatase production and it affected mineralization. Now, we're not ruling out CPED1 or Wnt16. God forbid, there's been so many studies done on Wnt16 in bone, so clearly it's an important bone gene. But in the context of the cell in its state, we could rule in ING3. So coming to type 2 diabetes, and obviously we're at a diabetes meeting, what we've been doing is actually working as a collective at the Children's Hospital of Philadelphia and Penn, and we're delighted to be doing this and working closely with the portal folks. And there was 2 awards made. One of us was to us in Philly. And the idea being is that we're going to try and find this confluence of evidence for all those GWAS loci that are out there for the causal effector gene or genes at each locus. And so Ben Voigt, who is heavily involved in GWAS loci, the discovery of GWAS loci for type 2 diabetes, Andrew Wells, my collaborator, Casey Brown, heavily involved in GTEx, and myself, and we're doing this combination of chromatin capture and EQTLs. And then we're handing those leads off to our functional colleagues who are experts in terms of doing functional validation of leads in various different contexts. Now, the complication with type 2 diabetes, of course, is there's so many candidate cell types. There's beta cell, there's liver, there's adipose, and I would argue gut as well. So that's sort of our pipeline. And the other awardees were amongst those individuals that got out are these people, and we're Anna Gloin, Steve Parker, Melina Klausenitzer, Karen Mulkey, and we're working together, and we're actually pooling our resources together and really trying to come up with a scoreboard, if you will, for the different approaches that we're using to try and say we're very, very convinced by certain genes at certain loci. This is some of the first fruits of our work. This is what we've just done at Penn and Chop. Sean Su here, working with data that was principally generated in Klaus Kestner's lab. We asked the question, rather than guessing the cell type for a disease, can the disease tell us which cell type to look at? So if we integrate the GWAS data sets that are publicly available and do LD score regression and say, where do we see an enrichment of traits in these different cell types for pancreatic cells, acinar, alpha, and beta cells, what we see is satisfyingly that for type 2 diabetes, and by the way, red means significant, for type 2 diabetes, we see a significant enrichment in alpha and beta cells, but not acinar cells. Conversely, for pancreatic cancer, where acinar cells are the primary candidate cell type, we see an enrichment in acinar cells. And also we see some interesting observations like sleep. We find an enrichment of sleep loci across all the different pancreatic cell types, and that's quite striking for us. So it's this idea of saying we are, before we endeavor to do a variant to gene mapping, am I working with a relevant cell type to that GWAS data set? And again, we're in the midst of this paper being in peer review, but once that's reviewed, we'll make the data available, and that'll of course go straight to the portal. One data set I think we've sent to the portal, and if it hasn't, I apologize, but it's publicly available. It's hypothalamus. And so Sheridan Littleton, who's at the back there, and Matt Paul published this paper in Nature Communications recently. And the idea was, can we look at the evolution of an ESC-derived, an embryonic stem cell-derived hypothalamic cell, going from the ESC state to the progenitor to the full-blown neuron. And what we find is that when we compare the RNA levels with GTEx, it does indeed look like a good proxy for hypothalamus, given the best match was with GTEx hypothalamus tissue. And we did the same thing there. We looked for enrichment of different traits. This is less easy to understand because it's got different colors, but the point was we also saw an enrichment of sleep. We saw an enrichment of major depression disorder, height, BMI, bipolar, and age of menarche. So then we are motivated to then go and do variant gene mapping, given we saw that enrichment. But we didn't see an enrichment for type 2 diabetes in this setting, so we're not motivated to do it there. talking about sleep, and again, sleep is a huge risk factor. If you've got sleep dysregulation, it's a huge risk factor for developing obesity and for developing type 2 diabetes. So I think there's a very strong motivation to study sleep in the context of diabetes. And so we've been really excited about this study, which is NIH funded, where we've taken the insomnia loci, which I had nothing to do with. I mean, I wasn't involved in the discovery of those loci at all, and it was principally from the UK Biobank. And we then did promoter capture, capture C with a TAC-seq in neuroprogenitors, IPS-derived neuroprogenitors. We then got a gene list from that and decided to take that gene list and see if we can see an effect in model organisms, flies and fish. And the idea being sleep is so conserved, everybody has to sleep. Sleep, evolution has never selected sleep away. So there's something obviously very fundamental there, and can we find conserved genes between species? So sending our gene list down to Texas A&M, we have a collaborator there, Alex Keane. We found this in fruit fly that we sent. Now, again, we're probably throwing out genes that are genuinely involved in human sleep. What we're looking for is those really conserved genes. And the ones in red are the ones that really had an effect on sleep patterns and activity patterns in Drosophila. So we either got very long sleepers or we get short sleepers, but we definitely got sleepy flies. The question was, can we make those sleepy flies, so if they're sleepy flies, do we also get sleepy fish? And so this is some of the data from the Drosophila, and we saw various different metrics for this gene, PIGQ, for instance, where PIGQ really, if we knock this down, it affects activity in the Drosophila. And then Amber Zimmerman, who's a relatively new postdoc in the group, and I share with Alan Pack at Penn, a fellow Scotsman, we find that it is indeed affecting sleep dysregulation in zebrafish as well. So we think this is quite a good way of taking data, which will be available in the portal. And you can then, if there is a good model organism, you could take your candidate gene list and do some sort of triage and have high value targets in which to pursue. So overall, as I come back to this idea of confluence of evidence, when you're doing functional studies, it's a large commitment. It's many years of work. As Sheridan would testify, she's doing her PhD on one key locus at the moment. So you want to have very high confidence in going after that. And I think we're afforded that with the sort of data sets that are available these days. And so I think that's exciting. And I mean, and a confluence of evidence study that I co-authored yesterday, that just came out in Nature Genetics, is a study of NAFLD. We use this proxy trait, which is C-alt, and through various different approaches and variant gene mapping using EQTLs, using algorithms that are available, was there a confluence of evidence for a particular causal gene at these new loci. So the idea being is that the public domain can aid in that confluence of evidence approach, and the data, which is in the portal, is becoming richer and richer all the time, especially as people like myself are putting up data there if you want to use it, and the idea of actually helping inform and drive your experiments going forward. So I'd just like to acknowledge the many collaborators that we have, and I'd also like to do a plug that Sheridan is going to be giving a talk on the key locus that she's focusing on, given that confluence of evidence for this key locus, which is a childhood obesity locus, and she'll be talking about that on Monday. Thank you.
The goal of this particular session is for someone who may be new, who've never worked with the portal, how do you interact with it? How do you use it? What type of, from a different perspective of not a statistical geneticist who spends their time looking at loci, but really someone who has a basic biology background or, from an industry's perspective, how might you use the portal to make decisions and navigate the various tools? Because there's a lot in there. We're going to try and distill it to sort of some simple tools that might help you sort of, make some decisions and really navigate the data we've presented. So with that, I'm going to turn to Maria. Yeah. Oh, beautiful. Yeah. Here you go, Struan. Beautiful.
The question is related to the slide of the enrichment of GWAS signals in 3 cell types. Why type 1 diabetes has no enrichment in any of these 3 cell types? Very good question. Yeah, we're scratching our head on that as well. I think we need to do that in our bank of immune cells that we already also have generated data for, so like beta cells, T cells. And I would bet, once we do that, that we would see an enrichment there, but yeah, given it's an autoimmune disease. But yeah, I would have expected to have seen something in at least beta cells, but we did not.
Thanks. Okay, so I'm not actually going to give a comprehensive demo of the portal. I'm just going to talk about sort of the directions that we're going. Okay, so yeah, the goal of the portal is to just help researchers move from all these tons of data that you've just been hearing about to derive actionable knowledge from all those data. And you've seen a similar pathway to this before. We want to help people move from the causal variants found from GWAS and blend in different kinds of data and find the causal genes, causal pathways, and how they relate to disease. So and when the portal first started out 6 years ago, whoops, it's just, sorry, this is advancing. Okay. When it started out, we started with just GWAS. That our mission initially was just to aggregate and display GWAS, and that in itself was a big help because people couldn't necessarily gather large GWAS data sets themselves or interpret them, and so the portal helped them do that. But it soon became apparent that we needed many more types of data and methods to help people move all the way along this pathway to understanding the genes that are involved in disease and how they work. So that's what we've been working on over the past 6 years. And okay, so yeah, and so what I'll just talk to you about briefly today is a couple of directions that we're going in. First of all, adding more expert curated information to the portals, and also adding more data types and developing methods to integrate and summarize and visualize these data types. And of course, those last two things go together. We need, yeah, the more data types we have, the more we need to be able to integrate them and draw meaning from them, the confluence of evidence, as Struan was saying. As the portal matures, we've also realized that our users can be kind of broadly divided into 2 different categories. One type is maybe the wet lab biologist or clinicians, people who are not genetics or genomics experts. They don't want to, analyze large files of data. They may look at a genetic association and not know what the p-value really means. But their work could be helped by the results that are available in genetic and genomic data. So for them, we need to distill and curate these data and present summaries that they can understand, that are meaningful for their work. And then, of course, the other category is the power user, that people who really understand the data and they want to see all the details. They want to have as much data available as possible, and they need versatile tools so they can easily access and explore these data. So we're trying to serve, gosh, I'm sorry, this thing just keeps on advancing on its own, or maybe I'm just looking at it. We're trying to serve both of these communities. So let's, for the first one, expert curated information to help the non-expert geneticist user. So we've seen this already in the first 2 talks, that you have variant associations across a region, and there may be many genes there, and it's not obvious first which is the causal variant. It's not the tip of the skyscraper usually, and which gene does this variant affect in the region. And so in many fields, and this seems to be an increasing thing in genetics now, is that it's not, people don't just do a GWAS, they will then integrate lots of different kinds of evidence and come up with a list of causal genes that they think are the likeliest causal genes for that disease or trait. And there's a growing number of these, and different groups use different types of evidence to make these predictions. This list just has some of the types of evidence that people for the lists that we have in the portal now, the types of evidence that have been used to make those lists. So of course, genetic evidence, where are the variants? Are they coding variants that change the sequence of a protein? Are they associated with monogenic disease? And then genomic evidence, all the regulatory elements, open chromatin and so forth, EQTLs. Some of these methods use algorithms like depict or pops, which themselves integrate different types of evidence to come up with a score. And there is a lot of different criteria that people use, and most importantly, perhaps, these people who make the lists are experts and they've been working in the field for a long time and they know the literature, they know what's been done. So they can also draw on, just various experiments, maybe in model organisms or in cells, and they kind of know what is known about each gene and they can factor that into their predictions. So we feel that these lists are very helpful for the users who can't necessarily predict the affected genes themselves. And so we want to present as many of these lists as possible. And so we've got a few already in the portal. We have one for type 2 diabetes that was the first list that we incorporated from Anuba Mahajan and Mark McCarthy, who incorporated a variety of evidence. And we have a list for coronary artery disease effectors from the Cardiogram Plus C4D consortium. And we just recently added a T1D effector list that Kyle Galton and his group generated. And this is a picture of that. It's hard to see on this screen, but we display these in tables that are interactive. So you can search the table for your favorite gene, but also the default view gives kind of a summary of the evidence about the gene. And then you can open up each row. And this, I swear I'm not pressing this button, it's just advancing by itself. You can open up each row and see the detailed evidence that is behind each prediction, and there are also links to the original sources. So this is the T1D list, and you can see there are links not only to PubMed, but also to the evidence, the epigenomic evidence in CMDGA. So we are about to incorporate another list for T1D effectors that was predicted in Steve Rich's group. So it won't be the same as the one predicted in the Galton group. It'll be interesting to compare the two. They've used different types of evidence. We're also looking at incorporating some lists from kidney function. And I just heard Struan mention a couple of lists of causal genes. So our goal is to incorporate as many of these lists as possible for as many traits, so that we really want to do that. So if you've generated a list of predicted effectors, we want to know about it. So we think these are very useful. Okay, so let's see. Okay, sorry, I think the slide is out of order. But anyway, as I mentioned, we want to add both distilled summaries and more detailed data and interactive tools for the power users. So okay, this is another example of a distilled summary. Our region page, if you enter a gene name into the homepage of the portal and search it, you will go to our region page. This looks a little bit different because we've just modified this display, and this is going to be in our release, which is happening possibly today or very soon. So this is a display of associations across a region of interest, all the top associations for all these different phenotypes, which I'm sure you can't read on this slide, but they're all there. And so we distill and summarize the information in this way, first of all, by doing bottom line integrative analysis. So all of the associations for each phenotype are analyzed together in a procedure that we've worked out. It's based on metal that our colleagues at the University of Michigan have developed, and we're continuing to refine this process. So it's meant to give kind of the best bottom line p-value for every association, every variant, based on integrating all of the available association data. We also do LD clumping to simplify the associations at a locus. And we also, so the table below that graphic shows the lead variant for each clump, but you can also see all of the individual variants in the clump if you want to. So one more example of a distilled summary. is a tool that's also in development called the Huge Calculator. And this was developed by Peter Dornbus and Jason Flanick. And it's based on the same idea that genetics can be very informative for different kinds of research, but it's not really accessible. People who are not expert geneticists can't really evaluate how much genetics supports their hypothesis of the involvement of a gene of interest in a trait or disease of interest. And so the Huge Calculator is a way of giving a score to that evidence, and it looks at both common and rare variation and assigns a score. So here's an example where a gene is this is, I think, SLC16A11, and the evidence from common and rare variation is compelling for its involvement in type 2 diabetes, which is, we know that's correct. So that's a good positive control there. So this is just another example of a way to make genetic information accessible to people who are not expert geneticists. So now on the other side, it providing lots of detailed data and custom interactive tools. Of course, this depends a lot on our partner resource, CMDGA, because they collect all of the regulatory and epigenomic data and send it through the portal to the portal, and we can display it on the portal. So I'm not going to go through this whole slide because we've heard a lot about this before, but the variant effects, the effects of especially non-coding variants on gene regulation in a tissue-specific way are very important for understanding how variants work and how they're involved in disease. So we need to incorporate all of these types of data, and we're continually incorporating more. And so here's an example of, a new tool that we're working on called the variant Sifter, as a way to integrate all of these different data types and let people explore them. So this is an example, again, from the literature, a gene involved in type 1 diabetes, BOC2. And the authors, Steve Rich's group, found that, so this is the top panel is the T1D associations in the BOC2 region. And then with this tool, the variant Sifter, you can add annotations for epigenomic annotations like transcription factor binding sites, accessible chromatin, and from different tissues. So those are the tracks of the annotations from T cells. And you can also add variant gene link data that are predicted by various methods, including the activity by contact, ABC method. And so the bottom track shows that. And all these tracks line up. There's also a table, which I haven't shown, that lists every variant, its position, and the details of the tissues that these annotations are seen in. So it's a way of for variants of interest, just seeing whether they do lie in regulatory regions in tissues and cell types that are disease relevant. So you can, with this tool, you can stack up multiple different association plots. You can look at many different tissues and annotations. So this is a really versatile tool that we really is going to help people prioritize genes and variants for further study. And I think that's all I have to say. Are there questions before I hand it over to Parul?
Hi, I'm Parul, and I'm presenting on behalf of the CMDGA team. This is the demo of Common Metabolic Diseases Genome Atlas, or CMDGA. So Common Metabolic Diseases Genome Atlas, or CMDGA, is a functional genomic portal. This is the home page of CMDGA, and you can access the home page at cmdga.org. So as part of this resource, we have collected and processed sequence data from thousands of functional genomic assays and develop data sharing tools to enable data integration with the knowledge portal, which is our genetics counterpart. In addition, we have also developed visualization tools that are complementary to our data sets. We also have improved recording of data analysis pipelines and quality control measures for each of the data sets that we have in our system. So the key information that we have in CMDJ are genomic experiments and annotations. Experiments are assays performed by individual research groups, and examples of these include ATAC-seq, RNA-seq, HiChips, HiC, etc. Annotations are analytical distillation of genomic experiments generated using software tools. Examples of annotations are accessible chromatin region, chromatin state, gene expression, target gene predictions, etc. In addition, we are also depositing new data types such as single cell embeddings, which are representation of intermediate files generated by single cell assays, genomic perturbation, which could be a single gene arrayed or genome-wide screen of perturbation in cell or animal model, and statistical models. Specifically, we are focusing on functional genomic models that help identification of target gene or region of interest. So how can you possibly use CMDGA to investigate information that you're interested in? So this is the home page. You can use the search tool. It's a very powerful search tool to search the entire data system. So here you can search by gene of interest, variant, study, or keyword of interest. So here I search for CFTR, which is a locus that has T1D causal variant RS779589. The result shows a combined GWAS study. It's a large-scale combined GWAS study and single-cell ATAC-seq in pancreas and peripheral blood cells. So this is the, so when you click on the result of that we get, you can visualize the single cell embedding for the ATAC-seq from the study. The information that you have is metadata that's linked to both the underlying, that has both underlying experiment and annotation for the study, which are gene expression level, chromatin peak in each cell type. We also provide, since this is a single cell embedding, we also provide the output file, which is the intermediate file in common H580 format, which can be used for further downstream analysis. So you can also visualize from the single cell study, you can also visualize the cell composition for the study in an interactive single cell genome browser, as well as you can look at expression of the gene of interest on a complementary gene expression browser. So in this case, we had looked at CFTR gene, and as you can see, the expression of this this particular gene is highly expressed in ductal cells. So then we also, the knowledge portal integrates the data from CMDGA and runs bioinformatic methods to integrate functional genomic annotations with disease risk. And here you can see the T1D risk variant RS7795896 is in enhancer region and an accessible chromatin region, and it reduces this particular risk variant, which is like at the top of the Manhattan plot, reduces the enhancer activity. So based on this, what we can conclude is all the investigation that we have done so far, what we conclude is the T1D risk variant RS7795896, while it reduces the enhancer activity, it increases the gene expression of CFTR gene in ductal cells. So this is how you go from a gene or locus of interest to interesting biological insights using CMDGA and the knowledge portal. In addition, for each of these assays, we have detailed data standards and QC metrics that were used for generation of the data and the analysis pipeline, as well as we have detailed information on the underlying analytical pipeline, the workflow, and the publication. So you can download data directly from CMDGA, as well as we provide data that's in CMDGA to the knowledge portal, where they run interesting analysis, bioinformatics analysis to intersect the risk variant with the genomic annotations and display it on their genome browser. So this is the CMDGA team. Kyle Galton is the principal investigator at CMDGA. Ying Sun is the data manager. She's here. And I'm Parul, I'm the software developer. We work closely with the knowledge portal team, as well as the AMP CMD consortium. So I can take any questions. Thank you, Melissa.
We're anticipating more single-cell data that will be quite informative for functional annotation coming in the CMD portal over the next few years. And I'm wondering if you could give us a sense of how you think those single-cell visualizations may change as the data sets grow. For example, visualizations in normal tissue versus disease, or the ability to look across experiments to see some differences in the single-cell data.
So we are integrating the available tools, publicly available tools, into our system. So how you can compare between normal and disease tissue cell type is we will have interactive filters where you can select a gene or peak of interest on the single cell browser, and then you can visualize change expression level on the single cell browser between disease and normal tissue, or if it's a ataxic, single cell ataxic model, you can also visualize difference in accessible peaks between different cell types on the browser. So that's an upcoming feature. Yeah. Thank you.
Other questions? Is there anything in the chat? Okay, great. So one last. Oh, great, Alex. Yes. I happen to know this.
I like questions.
Let's see how many we can get. My question is more related to the structure of the software. So I noticed that the knowledge portal and the other portal is constantly communicated each other.
Yeah.
So I wonder, like, if they live in the same, it's regarding like how the data is stored. So I wonder if the structure of how your portal stores the data is similar to how the knowledge portal stores the data, and if there's a better way to communicate that data instead of using an API.
Carl, do you want to take that one? Or you want me to take that?
Yeah, so all the data that's stored is extremely secured. No one who's not meant to have access can have access, both at CMDGA as well as knowledge portal. But we transfer the data to them via secure REST API. So yeah, there is some level of data storage replication because we have two distinct data storage cloud buckets.
So I would echo what Paul says, but I think we designed it to be a federated model, and the idea that at the Broad Institute, we house the summary statistics and in many cases the individual level data for the genetic data, because that's something we're structured to do, and we do that quite well. But Kyle's team at UCSD does this better. They store the raw data for the genomic annotations because they built it off the ENCODE model, the same software platform they use, and that's really useful because many people in the field are used to that, and it's tried and true. It's something we can extend and make specific for common metabolic diseases. So they warehouse the raw data there. We warehouse the raw data at the Broad, but on the portal, you see them integrated, because really what the user in many cases needs are summary representations of those data integrated in a format that's presented in an intuitive way. But if they want to drill down into the actual data, we're creating better connections. I think it's fair to say that, right now, if you're on the knowledge portal, getting to CMDGA is a bit of a path. But the idea being is that, we're going to present that in a more lightweight manner as we go forward to allow you to navigate between the two. So, it's a process, but it's designed that way so that actually you can allow other sites that have data sets. For example, the single cell atlas, it's built at the Broad as well, but it warehouses different data, but we want to make that accessible. So we don't want to reinvent the wheel in terms of a software platform, but we want to sort of modularize the portal such that you can display that data interactively. So it feels seamless, but the residing home of the data is different. Yeah.
Yeah, again, to emphasize, just in one line, we have the raw data, raw annotations, whereas the knowledge portal has integrated information, the integration of epigenetics and genetics, both combined together.
And ideally, if there's other software resources that are doing data types better than we are, that this creates an environment such that they can collaborate with us to make their data accessible to the portal in novel ways. That's our hope. Any more questions? Yeah, please, yes.
Yeah, I remember that yesterday we were talking about like how for different labs, they need to have their data in different places, federated nodes. So I wonder if you have a plan already of how we can put the data of each individual in a different place, and they are the only ones who can access to that data. And then you have the knowledge portal where you can see the summary if they allow it to make it public. Like, it's like having different instances of the knowledge portal. So each one has access to their own data. And then there's an option. So if they want to make it public, that data, other people can access to that data.
So you are tapping into our aspirational goals, which is great. Thank you. So that is actually really a direction we want to take. So the platform itself, right now is sort of, this is like, fully integrated. But what we've been building is a research platform. We call it BYOR, or Bring Your Own Results, which allows users, which is navigable directly from the portal. Actually, if you go down to scroll down to the bottom, you'll see that you can get there from in one of our homepages that describes the resource right there. Other knowledge portals in the network. If you click on that, Pearl. Yep, perfect. I'm gonna bring you to the page that has all the other portals in the knowledge portal network, but also a research platform for you to bring your own results. But I guess this is the beginning of the concept that you were saying, Alex, is other people have results. this is about results, not underlying data. So I'll get to that first. So results is someone has, for example, effector gene predictions or EQTL results that they want to make accessible. We have a mechanism now for them to actually use our software system to visualize and represent those data. They can keep the data where it is, but they can present them on a UI that they can make private. So just for example, if they're going to submit it for publication, they can have a private URL that they submit to a peer-reviewed journal that you can view. And then when it wants to be accessible, basically click a button and make it open access to the world. But the mechanism is also there for them to say, I want to contribute this data to the knowledge portal because it's relevant to disease X that you study. And that is it's already in the system, it's already harmonized, it's already processed in the way that we standardize and visualize, and boom, it can be on the portal. Raw data is more challenging, right? Because raw data has ethical restrictions and data use restrictions. So in many cases, you have to federate and have it such that the institution or the home domain of the data has to stay there and you can run compute on it. So thankfully, our colleagues at EBI and in Europe have built a mechanism where they can actually run the exact same software pipeline that we run at the Broad to process raw individual genetic data specifically and make it accessible in the portal in a way that you have no idea where it came from. It's a little bit of magic. But what's nice about that is that federated node can be portable to other institutes, for example, in different parts of the country, in different parts of the world, where data cannot leave. And that's another possibility, but you need to have, critical mass for that, because that is a software platform. It does require engineers who are intrepid enough to take that software and implement it at another institution. But that's something we're very keen to collaborate with others to do. Hopefully that answers your question. All right, fantastic. Actually, it's okay to end early because I love that. And thank you for dealing with all our technical challenges, those of you online. Imagine you are chuckling as we go. But I want to thank our speakers. I want to thank Melissa. I want to thank Struan. I want to thank Maria and Parl. It's great to work with great people and also great scientists, and the great people always comes first. So I thank you, and I thank our audience. And please use the portal. Send us feedback. It's actually, if you go back, Parl, would you mind do me a favor? Parl's doing this awesome moving along, to go to the homepage of the portal. A couple things I want to point out before we break. If you have any questions or feedback, you can actually go up to the top bar and you can contact us. And this comes to actually to some of the people in this room. And we respond to you within 24 hours. If you have any feedback, if you want to be added to our webinars or our focus sessions, or you just have a question, or you want to actually contribute data, please contact us. I take this very seriously, and I will say that we do a really good job of this. So we'll follow up with you. Other things to think about, not just in common metabolic diseases. This is a challenge across all of common disease. So we're very lucky that we've worked with other communities to build knowledge portals in lung disease, musculoskeletal disease, and for actually different organelles, which has been really fun. And that's an exploratory path for us in terms of how to represent portals in that space. And we're also expanding to all of common diseases with Association to Function Knowledge Portal coming in September that will tackle common disease, the same software platform with the same visualizations across the spectrum of common disease. So look for that in the fall. And with that, I will conclude and thank you for your time and your attention, and enjoy the day if you're in New Orleans, and enjoy ADA, and take care. Thank you.